Hello all. So today we are talking about metabolism and function. This is part 2 of chapter 4. Here we go. First, let's look at digestion. When we consume carbohydrates, they are broken down mechanically and chemically in the digestive tract. This process converts the food into absorbable forms. Specific enzymes play a crucial role in breaking down carbohydrates into smaller substances that can be absorbed. For instance, amylase breaks down starches into sugars. Next, let's talk about absorption. The majority of carbohydrate absorption occurs in the small intestine, although small amounts are absorbed in the stomach and large intestines as well. This process happens through several mechanisms. Passive diffusion. This is where molecules move from an area of higher concentration to an area of lower concentration without the need for energy. Facilitated diffusion is where molecules also move from higher to lower concentration, but they require the help of a carrier protein. And then active transport. This is a process that requires energy to move molecules against a concentration gradient from lower to higher concentration, often involving specific transport proteins. And finally, let's touch on excretion. The remnants of carbohydrate digestion that are not absorbed along with other waste products are excreted from the body. This process helps maintain balance and remove waste efficiently. The alimentary canal, also known as the digestive tract or gastrointestinal tract, is a long hollow tube that extends from the mouth to the anus. It's responsible for the digestion and absorption of food and nutrients. The major organs of the alimentary canal include the mouth, the pharynx, esophagus, stomach, small intestine, large intestine, which is the colon, and anus. Each organ plays a specific role in the breakdown of food, the absorption of nutrients, and elimination of waste. The villus is a key structure in the small intestine that facilitates nutrient absorption into the bloodstream. Some of the main components are the epithelial cells that cover the villus surface and contain specialized cells like enterocytes. The lamina propria, which is a connective tissue containing blood vessels and lymphatics for nutrient support, and muscularis mucosa. It's a thin layer of smooth muscle supporting villus movement. The microscopic features are the microvilli, which are tiny projections on enterocyte surfaces that increase surface area for absorption, and capillaries, which are small blood vessels in the lamina propria where absorbed nutrients enter the bloodstream. The lacteals are specialized lymphatic vessels absorbing fat-soluble nutrients into the lymphatic system. So the function of the villus is really to absorb nutrients such as sugars, amino acids, fatty acids, vitamins, and minerals. And its key role is in maintaining the body's nutritional balance and overall health. So understanding the villus structure is really crucial to comprehending nutrient absorption processes in the digestive system. Here's a structured explanation of how dietary carbohydrates are digested and absorbed, along with implications for sports performance. There's enzymatic activity with amylase, which is secreted by salivary glands and pancreas. Amylase breaks down complex carbohydrates, which are starches, into shorter polysaccharides and maltose in the mouth and small intestines. Now that absorption process takes place in the small intestines where most carbohydrate digestion occurs. And so the monosaccharides, the enzymes on the surface of the enterocytes, which again are the specialized cells in the villi of the small intestine, further break down maltose, sucrose, and lactose into monosaccharides, which is glucose, fructose, and galactose, and transport monosaccharides are absorbed into enterocytes and then transported across the cell membrane into capillaries of the villi, where there the monosaccharides are transported via the bloodstream to the liver and then distributed throughout the body for energy. Now, some implications for sports performance are sports drinks and carbohydrate absorption. So the target is the targeting monosaccharide receptors. So sports drinks formulated with specific ratios of glucose and fructose can utilize different transporters in the villi, potentially increasing carbohydrate absorption rates during exercise. And for enhanced water absorption, so including sodium in sports drinks can enhance water absorption via co-transport mechanisms in the intestines, aiding in hydration during prolonged exercise. And then the gastric emptying and water absorption. Impact on performance is the rate of gastric emptying affects how quickly fluids and nutrients, including carbohydrates, enter the bloodstream. Optimizing this process can help maintain energy levels during exercise. And of course, the hydration balance. Sports drinks with appropriate carbohydrate concentrations and electrolytes can promote faster gastric emptying and fluid absorption, supporting hydration strategies in athletes. So high concentrations of simple sugars, there's a dumping syndrome. And so consuming high concentrations of simple sugars like glucose and fructose may overwhelm the digestive system, leading to discomfort, bloating, and potentially impacting performance negatively. And then lactose and digestive sensitivity creates gas production. So some athletes may experience gas production when consuming lactose containing sports drinks or foods, which can be uncomfortable and affect performance, particularly in endurance sports. Once dietary carbohydrates are absorbed into the body, they undergo several processes. The first of those is a conversion to glucose. And so there's a glucose formation. Most absorbed carbohydrates are converted into glucose, which is the primary form of sugar circulating in the bloodstream. And that gives us immediate energy for the cells throughout the body, especially important for muscle function during exercise, and the regulation of blood glucose. When blood glucose levels rise after a meal, the pancreas releases insulin, which facilitates the uptake of glucose into cells for energy or storage. And with an excess glucose, it is converted into glycogen in the liver and muscles for short-term energy storage. The effects on blood glucose levels, we look at the glycemic index, or GI. This measures how quickly and how much a carbohydrate containing food raises blood glucose levels compared to pure glucose. Low GI foods that release glucose slowly into the bloodstream, promoting more stable blood sugar levels over time, are considered to be low GI. And this would be whole grains or legumes. High GA foods are foods that rapidly increase blood glucose levels, such as white bread or sugary foods, which can be beneficial for rapid energy replenishment, but may cause spikes and crashes in blood sugar levels. And then the glycemic load. Now, to calculate the glycemic load, it takes into account both the quality, which is the glycemic index, and quantity of carbohydrates consumed. Now, the impact on health is a lower glycemic load diet may help manage blood sugar levels and reduce the risk of chronic diseases like diabetes and cardiovascular disease. As far as practical implications for sports performance, for pre-exercise fueling, you want to choose carbohydrates with a moderate to high GI and GL, so glycemic index and glycemic load, that can provide quick energy before exercising, promoting optimal performance. During exercise, you want to consume carbohydrates with a high glycemic index and glycemic load during so that prolonged endurance activities can help maintain glucose levels and delay fatigue. Now, post-exercise recovery, you want to include carbohydrates with a moderate to high GI and GL, and post-exercise meals or snacks that can replenish glycogen stores more rapidly. Understanding the glycemic index and glycemic loads of foods allows athletes to tailor their carbohydrate intake to optimize energy availability and maintain stable blood glucose levels throughout training and competition. The glycemic index, again, is a ranking system that measures how quickly carbohydrates in food raise blood glucose levels compared to pure glucose, which is assigned a GI value of 100. Here's a breakdown of the GI rating scale. So high GI foods have a GI of 70 or more. These foods cause a rapid increase in blood glucose levels after consumption. Examples, again, include white bread, white rice, and most sugary foods. Medium GI foods have a GI between 55 and 69. These foods raise blood glucose levels moderately compared to high GI foods. And examples include whole wheat products, brown rice, and some fruits like bananas. Low GI foods have a GI of 55 or less. These foods cause a slower and more gradual rise in glucose levels. Examples include most vegetables, legumes, whole grains like oats and barley, and some fruits like apples and berries. The GI scale provides valuable information for athletes and individuals seeking to manage their blood sugar levels and optimize carbohydrate intake for energy and performance. The glycemic load expands on the glycemic index by taking into account both the quality, which is the glycemic index, and quantity of carbohydrates consumed in a serving of food. The GL is calculated using the following formula. So glycemic index times the grams of non-fiber carbohydrates in one serving divided by 100, where GI is, of course, the glycemic index of the food, and then the grams of non-fiber carbohydrates refers to the total amount of carbohydrates in the food minus the dietary fiber content. Now, high GL foods have a GL of 20 or more, and these foods can cause a significant spike in the blood glucose levels. And again, we're talking about white rice, white bread, and sugary snacks. The medium glycemic load foods have a GL between 11 and 19 and moderately raise blood glucose levels compared to high GL foods. Again, examples are whole wheat bread, oatmeal, and some fruits like grapes. And then low GL foods that have a GL of 10 or less. These foods have a minimal impact on blood glucose levels and promote more stable energy levels. Examples, again, are vegetables, legumes, and whole grains like barley and quinoa. And this table just shows the glycemic index and glycemic load of most common foods. It's a great resource for you. The metabolic fate of blood glucose involves its regulation and utilization throughout the body, influenced by factors like dietary intake, insulin secretion, and overall metabolic needs. So normal blood glucose levels range typically between 80 to 100 milligrams per deciliter or 80 to 100 milligrams %. Stable levels are really important crucial for providing energy to cells, especially the brain and the muscles. Now, effects of high GI foods, as one of those is hyperglycemia, which is based upon the consumption of high GI foods that lead to a rapid increase in blood glucose levels of greater than 140 milligrams per deciliter, which is also known as hyperglycemia. Now, the insulin response, or the response to elevated blood glucose levels, is really the pancreas secreting insulin to facilitate the uptake of glucose by tissues for energy or storage. Now, insulin's role is, again, for glucose uptake. It enables glucose to enter cells where it can be used immediately for energy or stored as glycogen in the liver and muscles. It's Insulin also inhibits the breakdown of glycogen into glucose, or glycogenolysis, and promotes the conversion of glucose to fat for long-term or energy storage. Now, the risk of hypoglycemia is low blood glucose, and so excessive insulin secretion or insufficient carbohydrate intake can lead to hypoglycemia, which is blood glucose that levels below 70 milligrams per deciliter, which can cause symptoms like dizziness, confusion, and in severe cases, unconsciousness. So if we look at glucose transport with insulin, you'll see that insulin binds to its receptor on cell membranes, initiating a signal cascade that results in the translocation of GLUT4 transporters from intracellular vesicles to the cell surface. There's also an increased transport. So GLUT4 transporters on the cell surface facilitate the uptake of glucose from the bloodstream into the cells, particularly in skeletal muscle, cardiac muscle, and in adipose tissue. Now, the metabolic effects are energy production. So inside the cell, glucose undergoes glycolysis to produce ATP, which is the energy currency of cells, and storage. So excess glucose is converted into glycogen in muscles and liver for short-term energy storage, or into triglycerides in adipose tissue for long-term energy storage. And then regulation, the regulation of blood glucose levels. Insulin helps regulate blood glucose levels by promoting glucose uptake into the cells, thereby lowering blood glucose concentrations. And insulin promotes an anabolic process, including protein synthesis and cell growth, by providing glucose as a substrate for building macromolecules. Now, if we look at glucose transport without insulin or with insulin resistance, we'll notice that there's a reduced GLUT4 activation. And so without sufficient insulin signaling, such as in insulin resistance or deficiency, GLUT4 transporters do not translocate efficiently to the cell membrane. And there's a decreased uptake in glucose into the cells, particularly in insulin-sensitive tissues like muscle and fat. So the metabolic effects of that are impaired energy production. Cells are less able to use glucose for energy production, leading to a reliance on alternative metabolic pathways, such as fatty acid oxidation and ketone production. And high blood glucose levels remain elevated, contributing to metabolic dysfunction and potential complications, so hyperglycemia. There are clinical implications to that, as you probably know, type 1 diabetes, as a result from a lack of insulin production, leading to uncontrolled hyperglycemia and the need for exogenous insulin therapy. And type 2 diabetes, which is characterized by insulin resistance, where cells become less responsive to insulin, requiring strategies to improve insulin sensitivity through diet, exercise, and medication. The fates of blood glucose once it enters the body includes several metabolic pathways. Glucose as a primary source of energy for cells throughout the body, especially vital organs like the brain and muscles. Through glycolysis and subsequent pathways in cellular respiration, glucose is converted into ATP, which is adenosine triphosphate, which powers cellular functions. Now, excess glucose beyond immediate energy needs is stored in the liver and muscles as glycogen. Now, glycogen serves as a readily accessible source of glucose for energy production during periods of fasting or increased energy demand, such as exercise. Now, when glycogen stores are full, surplus glucose can be converted into fatty acids through lipogenesis in the liver. These fatty acids are then esterified into triglycerides and stored in adipose tissue for long-term energy storage. In conditions of severely elevated blood glucose levels with hyperglycemia, such as uncontrolled diabetes, the kidneys may excrete excess glucose into the urine. This condition is known as glucosuria and is indicative of poorly controlled blood sugar levels. So how much total energy do we store as carbohydrates? You'll see it is expressed as millimoles. So 1 mole of glucose is 180 grams or about 6 ounces, whereas 1 millimole is 1/1000th of 180 grams or 180 milligrams. So you'll see the glass of milk as 1 mole of glucose, which is equal to 100 grams. It's 6 ounces. That means it's 36 teaspoons. Now, if we put that into millimoles, 1 millimole of glucose is equal to 180 milligrams, and as 6/100,000ths of an ounce and 36/100ths of a teaspoon. The total body storage of carbohydrates in the form of glycogen amounts to a limited energy reserve. So, in estimating that glycogen storage capacity, blood has a very limited capacity to store carbohydrates. You'll see it's 5 grams, and so equivalent to about 20 kcal, versus the liver, which can store approximately 100 grams of glycogen. And so if we multiply, again, we know 4 kcal per gram for carbohydrates. That would be about 400 kcal. And with muscle, the average adult has approximately 400 grams of glycogen stored in their muscle. And so 400 grams times 4 kcal per gram is equal to 1,600 calories. So you take that energy and calories and you look at liver glycogen and muscle glycogen, you multiply that by 4. So 400 kcal in the liver plus 1,600 kcal in the muscle, that equals about 2,000 kcal. Given that 1 gram of glycogen is associated with approximately 3 grams of water, the actual storage is a little less due to the water weight associated with glycogen storage. So the body primarily uses these glycogen stores for energy. And after the stores are depleted, the body shifts to using fat and protein or muscle tissue for energy. In the case of 1 full day of starvation, it does deplete it very quickly. So when carbohydrates are not available, the body can produce glucose and by utilizing gluconeogenesis. So the body will start producing glucose from non-carbohydrate resources, such as amino acids and glycerol, to help maintain blood glucose levels. In protein metabolism, it's the glucose-alanine cycle process, and this is where muscle protein breaks down into amino acids, such as alanine, which travel to the liver. The liver converts alanine into pyruvate through a transamination reaction process. That pyruvate enters the gluconeogenesis pathway, leading to the production of glucose. Now, the glucose-alanine cycle is a major mechanism for transferring nitrogen from muscle to the liver for excretion and for generating glucose from muscle protein during prolonged fasting or exercise. The efficiency of glucose production from alanine is variable and depends on several factors, including the individual's metabolic state and the availability of other substrates for gluconeogenesis. However, it is generally estimated that approximately 58% of alanine carbon can be converted into glucose, which is about 0.56 grams of glucose from 1 gram of protein. Now, fat metabolism, glycerol is a component of triglycerides, which accounts for approximately 10% of the triglyceride molecule by weight. And so glycerol can be converted into glucose through the gluconeogenesis pathway in the liver. Approximately 1 gram of glucose can be produced from 1 gram of glycerol. This makes glycerol a relatively efficient source of glucose compared to protein, and particularly during periods of fasting or when carbohydrate intake is limited. While glycerol can contribute to glucose production, the primary source of glucose during fasting is gluconeogenesis from amino acids, particularly from muscle protein breakdown. Now, pyruvate produced from glycolysis can be used to generate glucose as well. And during anaerobic metabolism, lactate is produced by muscles and transported to the liver, where it can be converted back into glucose through the Cori cycle. These pathways are crucial, especially during fasting and intense exercise or in low carbohydrate diets, to maintain blood glucose levels and supply energy to glucose-dependent tissues like the brain and red blood cells. So major functions of carbohydrates in human nutrition is to supply energy. It is the body's primary energy source. And so carbohydrates are quickly converted into glucose, which fuels cellular activities. It's really essential for brain and central nervous system and red blood cells. These tissues rely heavily on glucose for proper function. The brain, for instance, consumes approximately 120 grams of glucose per day. It's also important fuel for exercise and other functions. The component of glycolipids and glycoproteins. Carbohydrates combine with lipids to form glycolipids, which are essential for cell membrane stability and cell recognition processes. Ribose in RNA is a 5-carbon sugar, and it's a crucial component of RNA, or ribonucleic acid. RNA is involved in protein synthesis and various other biological roles as well. The metabolism of carbohydrates, fats, and proteins are intricately interconnected, ensuring a constant supply of energy and essential building blocks for the body. So glucose is the central hub, and carbohydrates provide the primary fuel source for most cells, particularly the brain and red blood cells. Fat, on the other hand, can be broken down into fatty acids and glycerol, and that glycerol can be converted into glucose through gluconeogenesis, which you see on the right-hand side of the diagram here. Now, protein can be used for gluconeogenesis as well, especially during prolonged fasting or starvation. Now, the interconversion of these energy stores, the carbohydrates, excess carbohydrates, are stored as glycogen in the liver and muscles, where fats, the excess carbohydrates and proteins, can be converted into fatty acids and stored as triglycerides in adipose tissue. Now, proteins are primarily used for building and repairing tissues, but they can be broken down for energy in times of need. The intricate interplay between carbohydrate, fat, and protein metabolism ensures a constant supply of energy and essential nutrients for the body. Understanding these interrelationships is crucial for promoting health and preventing metabolic disorders. And so key concepts to this part 2, metabolism and digestion, as most ingested carbohydrates are initially converted into blood glucose and used for energy or stored as liver and muscle glycogen. But excess carbohydrate may be converted into fat. Now, the glycemic index is a measure of the rate of digestion and absorption of 50 grams of a carbohydrate food and the resultant effect on the blood glucose level. The glycemic load is similar, but it's based on the typical serving size for carbohydrate food. And so there are different numerical rankings used for GI and GL. The 3 sources of carbohydrates in the body of an average adult male are blood glucose, which is about 5 grams which is like 20 kcals, liver glycogen, which is 75 to 100 grams, and then muscle glycogen, which is about 350 to 400 grams on 1,400 or 1,600 calorie diet. The liver is the main organ that can produce glucose from certain byproducts of protein and fat, which is a process known as gluconeogenesis. The major function of carbohydrates in human metabolism is to supply energy. Blood glucose is essential for optimal functioning of the nervous system, whereas muscle glycogen is essential for both anaerobic capacity and aerobic power exercise. And I thank you for joining me today. We will be going into it's part 3 of chapter 4. I'll talk with you soon. Have a great day.